Hello everyone, this is Kishore Kumar. Today I am going to discuss about the pentose phosphate pathway. So this pentose phosphate pathway will have a multiple names, but a very common name is pentose phosphate pathway. And we should learn about the different reactions, different enzymes which can be involved in this particular pathway. We're going to look into coming slides. And you should be remember this because the glucose should have the two oxidative pathways. So one oxidative pathway is to be involved for the generation of ATP that we already discussed in previous classes. That is involvement of glycolysis, Krebs cycle, electron transport chain, and oxidative phosphorylation, and finally the ATP is synthesized. Right, that is the one way of the oxidation of glucose, and there is another way that is also the oxidation of same glucose. So this particular the other pathway, oxidation of glucose pathway, we call pentose phosphate pathway. So you should be remember that even though these two are an oxidative pathways, there is a clear difference. So the clear difference we should understand here, first one is the first pathway which is related with the ATP generation, that is generation of cellular energy. So this generation of cellular energy, which is mostly related with the catabolic pathway. So this is we call catabolic pathway of glucose. But if you see this pentose phosphate pathway, this is instead of generating ATP, which can be involved for the synthesis of certain biomolecules. Synthesis of certain biomolecules, which can be from the intermediates of this pentose phosphate pathway. So we call this pentose phosphate pathway is to be an anabolic, whereas the other pathway that is to be an catabolic. It's clear? So now we will going to discuss it, and you try to remember this pentose phosphate pathway and Hexose monophosphate shunt and phosphogluconate pathway. These are the three alternative names which are all related with a single pathway. So we will discuss in this particular class. We are going to discuss about the few important things which are related with this particular pathway. Here, first of all, I am going to give you a few introduction about this particular pathway, about their occurrence, and about their different natures. That all I'll discuss in a single slide. That is an introduction. After that, I'll discuss about the reactions of this particular pathway and the enzymes which are involved in the particular reactions, and even the regulation part also. That I'm discussing in the second concept. And third one, this is to be important because why this pentose phosphate pathway is to be important for the cell. In that particular scenario, I'm going to discuss about the significance of this particular, the significance of these pathways. And last, coming to the disorders, is there any disorders that are associating with this particular pathway? That is also I'm going to discuss in this particular session, right? So viewers, you just remember that. It's all clear because the pentose phosphate pathway, if you take a basic example, this is an alternative pathway of glucose oxidation. This we should remember. So as I discussing the in my previous slide, so you should be remember that this is to be an alternative pathway of glucose oxidation, where exactly ATP is not synthesizing. Instead of ATP, there are other products which are to be synthesized. So if you take this particular pathway, this is more we call anabolic in nature. So anabolic in nature means we should be understand that this particular pathway involved in biosynthesis pathway or biosynthetic mechanisms. So biosynthetic mechanisms will come with the example of anabolic nature. So where does it exactly happen? This can be happening in the cytosol of these cells. So what kind of the cells which are actively involving in this particular pathway, that particular thing which I'm giving the examples in the coming points, but you should be remember that this can be operated in the cytosol, and you take the byproducts, or we call itself products, or intermediate products. Intermediate products of pentose phosphate pathway are majorly two products, which are NADPH. Second one is pentoses. So the pentoses and NADPH, which are to be having an enormous applications or enormous uses to the cell, and at last we will discuss in a significance what are the importance of this NADPH and pentoses, and you can see this particular pathway which is very unique and multifunctional pathway. Why I am saying unique? Because the byproducts which are formed in this particular pathway will be involved in various kind of reactions. So these various kinds of the reactions, so that are very unique to this particular pathway, and you can see this where exactly this particular pathway is activating. So especially if you see the tissues that are actively involving in the fatty acid and steroid metabolism. If you take any particular tissues, if they are active in the fatty acid and steroid metabolism, where exactly this pentose phosphate pathway is very active. Here I have given the best example. You can see here the liver, adipose tissue, adrenal gland, RBC, testis, and lactating mammary glands. So these are the essential tissues where exactly this particular pathway is very active. So this we should remember, because this particular fatty acid metabolism which is required reducing equivalence. So these reducing compounds are nothing but NADPH, which is essential for this pathways. So that these are the tissues which are actively engaging these pathways. So definitely this can be involved in the major portion of the synthesis of pentose phosphate pathway. And you can see in this pentose phosphate pathway should have the for the better understanding, we are divided into two phases. So this is two phases, right? The two phases, one phase we call oxidative phase, and second phase we call non-oxidative phase. The oxidative phase and non-oxidative phase, these are to be involved together. These are involved in pentose phosphate reactions. So first reactions are coming oxidative phase. The oxidative phase will be involved in these reactions. You can see up to ribose 5-phosphate from the glucose 6-phosphate. So majorly you should understand what is oxidative phase. On oxidative phase, there will be two products which are formed. One is to be NADPH, second one is to be a ribose 5-phosphate. These are the two products which are formed in the oxidative phase, and these are having a different kind of the uses into the cells for the different purposes. So if you see the NADPH, the NADPH is nothing but, so this is the structure which is related with the NADPH, and the NADPH which is important for the various kinds of the cellular functions that we will discuss. And if you see the ribose 5-phosphate, a ribose 5-phosphate structure will be like this, and this is always to be, we will write it in a cyclic structure. But better you can understand what is the cyclic structure and open structures and where it can be predominantly existing. You just go back to my carbohydrate videos, and you can better understand this, the structural conversion of cyclic to the linear to the cyclic. It's clear? But these are the two important products which are to be formed in oxidative phase. Then come to the non-oxidative phase. This non-oxidative phase, you should remember that this is only involved for the involving for the regeneration of glucose 6-phosphate, right? So the regeneration of glucose 6-phosphate is to be taken place in the non-oxidative phase, where exactly the enough amounts of glucose 6-phosphate is to be present. This is the initial substrate, so that this particular pathway is continuously happening in that particular tissues where the fatty acid metabolism is more. Clear? So this is what this particular reaction pathway will have the two different phases, oxidative and non-oxidative phases. And if you come to actual reactions, what are happening in pentose phosphate pathway? So here I showed a very easy understanding diagram, because here I have not put all these structural figures, because that is not to be important to learn, but the concept, what are the products, what are the substrates which can be involving in this particular pathway is to be important. So you try to understand this. I'll try to explain in a simple manner. You don't bother about the structural aspects of this particular products, right? So if you see this, the first pathway is the glucose 6-phosphate. The glucose 6-phosphate is the initial substrate for this pathway. This can be converted into the 6-phosphogluconolactone, right? But here you should be remember that something which is written in the red color, something which is written in the blue color, or something which is written in the blue color, that you should be understand. The red color is nothing but the number of molecules which are involving. It's clear? So here 6 glucose 6-phosphates are involving. Then what do you mean by green color? The green color is nothing but that is the carbon length of the particular molecule. For suppose if you take glucose is having the 6 carbons. So the carbon length or the carbon number is given in the blue color, and the total number of molecule which are involving, that is given in the red color. So you can see entire the reaction which will be represented in the same manner. So 6 glucose 6-phosphate molecules are converting into 6-phosphogluconolactone molecules. For this, the enzyme is involved is dehydrogenase, glucose 6-phosphate dehydrogenase. Whenever you see the dehydrogenase, which already I discussed in earlier videos, so dehydrogenase definitely that can be involved for the synthesis of NADPH or synthesis of reducing equivalence. So here 6 NADP plus is converting into 6 NADPH. So this is each NADPH is formed for the each glucose molecule. So total 6 NADPHs are formed, right? So after this reaction, this is to be on next conversion is 6-phosphogluconate. This can be done with the help of lactanoase, and where the 6 water molecules are utilized. And again, this can be converted into ribulose 5-phosphate. This ribulose 5-phosphate is again should require the dehydrogenase. But this dehydrogenase and the first dehydrogenase enzyme both are different, but their function is to be same. So here also again 6 molecules of NADPH is formed. Then after that, the ribulose 5-phosphate, which is to be interconverted because of isomerases, and the 2 molecule of ribulose 5-phosphate and ribose 5-phosphate. These 2 molecules will condense and will form the pseudoeptylose 7-phosphate and glycerol 8,3-phosphate. At this particular stage, you should remember, this particular stage, the enzyme which is involving is transketolase. So whenever there are 2 transketolases that can be involved in this particular pathway, one is to be here and second one is to be the next reaction which I've showed. So whenever you see the transketolase enzyme activity, it should require the TPP cofactor, that is thiamine pyrophosphate. During this particular reaction, 2 carbon molecule is attached to the TPP. TPP act as a 2 carbon carrier, and finally it can donate to the products. So this is how the 2 carbons are initially taken from the substrate, and it can be given to the products. So this is how the TPP, thiamine pyrophosphate, is involved for the transfer of 2 carbon molecules. So after this, then these two more products will combine together and will form these two. These are erythrose 4-phosphate and fructose 6-phosphate. Here the enzyme which is required is transaldolase. And again, you should be remember this xylose 5-phosphate and erythrose 4-phosphate, which can be converted into fructose 6-phosphate and glycerolide 3-phosphate, where exactly again transketolase is involved. And the ultimate aim of this particular up to this part is to be oxidative phase, as we discussed in previous slide, and the rest of the part is to be non-oxidative phase, where exactly the importance of non-oxidative phase is to be produce the glucose 6-phosphate. Clear? The glucose 6-phosphate is to be produced at the end of the non-oxidative phase, and this level is to be maintained, and it can again going to one more pathway of pentose phosphate. It's clear? So here, what are the different kinds of the enzymes which are involving? Of course, this is to be reversal of glycolysis. You can see the fructose 1,6-bisphosphatase enzyme, phosphofructohexose isomerase enzyme that can convert fructose 6-phosphate to glucose 6-phosphate, and so on. So finally, it can give the 5 molecules of glucose 6-phosphate. This we should be remember. Initially, 6 molecules of glucose 6-phosphate is involved, and later on, only 5 molecules are regenerated. So 1 molecule of glucose is to be utilized in this particular pathway. Clear? If you see the overall reaction of this pentose phosphate pathway, that where exactly 6 glucose 6-phosphate, 6 molecules of glucose 6-phosphate is involved, plus 12 NADP plus involved, plus 6 water molecules are involving. So these substrate is converting into the 6 CO2 molecules, because here initially, which is 6 glucose 6-phosphate is 6 molecules of glucose 6-phosphate is involved, but finally 5 molecules of glucose 6-phosphate is to be generated. But the rest of the 1 molecule, the 1 molecule of glucose is nothing but the 6 carbons that are released as CO2. That is 6 CO2 molecules plus 12 NADPH and plus 12 H plus finally 5 glucose 6-phosphates are regenerated. It's clear? This is with the overall reactions that we can represent in the simple manner. So coming to the regulation. So the regulation, which is to be very important, because you can see that in any kind of the metabolism, that has to be regulated with the same or resembling pathways. This is to be very important. In this particular nature, if you see, because the glucose is converting into the glucose 6-phosphate, this glucose 6-phosphate will have the 2 ways. Either it can go into the glycolysis, citric acid cycle, finally ATP generation, or it can go into the pentose phosphate pathway, finally it can produce the NADPH and ribose. So the regulation between the glycolysis and this pentose phosphate pathway has to be coordinated. This is very important. The coordination will be like this. But suppose if you see this, if you take a cell, any cell, that is liver cell or mammary gland cell, that particular cell is having enough amount of ATP. So it should have the good amount of ATP in that particular cell. What does it happen? That ATP will be acting as an inhibitory to this oxidative pathway. So definitely, if the enough amount of ATP is present to the cell, definitely the energy giving oxidation pathway, that is glycolysis, Krebs cycle, ETC, and oxidative phosphorylation will stop, right? Then the product is glucose 6-phosphate is accumulating, and that is going into the pentose phosphate pathway. Clear? Then even the pentose phosphate pathway, definitely in between the products, that is NADPH, if it is to be more in a cell, definitely this will go and act on the pathway, and the dehydrogenase enzyme, that is glucose 6-phosphate dehydrogenase enzyme, and which will say that I am having enough amount of I'm having enough number of amount in particular cell, so you please try to stop for this particular pathway. This has information which is given by NADPH. This we call feedback inhibition. The feedback inhibition means where the end product will be involved for the regulation of that particular pathway, we call feedback inhibition. So if it is to be like this, and it can go forward to this pathway, right? These are coordinated. And you should be remember that even if you take the fatty acid metabolic tissues, where exactly this is to be, and 10% of glucose is to be converting into pentose phosphate pathway. That anyway we should be remember. If you see the significance, that's all related with the regulation and various reactions which are involving, and a few enzymes which are to be important for this particular pathway. It's enough to know only that much, right? Don't bother about the complex structural aspects of this particular pathway. And if you see the significance, so definitely the significance which is related with these two products. One is pentoses, second one is NADPH. If you see the pentoses, especially pentoses are derivatives of the pentoses will be involved for the synthesis of nucleic acids. So that is RNA and DNA. So the RNA and DNA, which is to be important for having of ribose, that is ribose 5-phosphate. Clear? So in this nucleic acid synthesis, these pentoses will be involved, and not only the nucleic acids, it can be also involved for the generation of various kinds of the products, that is ATP, NAD+, FAD, and coenzyme A. These are also be nucleotides, and which are not corresponding to the hereditary function, but even though which are to be important for the cells. So that you should be remember that this can be involved in a hereditary as well as non-hereditary functions also. It's clear? And you can see in the NADPH. In a NADPH, this is to be on reducing nature. So this reducing nature will have a various kinds of applications. Listen, here I am trying to discuss a very few basics which are related with the NADPH. First one, if you see the function which is involved in fatty acids and steroid synthesis, which I already discussed in a previous slide, right? And this is also be important for the synthesis of enzyme from the certain amino acids. That enzyme is glutamate dehydrogenase. So this particular enzyme also should require this NADPH. And the antioxidant reactions, these antioxidant reactions which are done by the antioxidant enzymes, and for best example is superoxide dismutase and glutathione reductase. There are many antioxidant enzymes that are present. For its activity, definitely the NADPH is present. But you should be remember that what is antioxidants or what is oxidant, because during many metabolic reactions, the reactive oxygen species that can be generated, the reactive oxygen species, if these are not taken into the neutralize, if these are not neutralizing, definitely this will go attacking on the cell membrane, DNA, and even proteins also, finally make them to be destroyed. So in that particular way, these antioxidants will be try to prevent that effect of reactive oxygen species. For that particular type of reactions, this NADPH is to be very important. And the detoxification of the drugs. So this mechanism, which is majorly done by the liver cell, that process we call genobiotics, where cytochrome P450 is a major enzyme that can be involved for the detoxification, which for its activity it should require this NADPH. And even phagocytosis, that is the engulfing of the any material from the any material by the cell, we call phagocytosis. There after engulfing, there will be many different kind of reactions which are to be initiating in the cell, that are all related in that particular reactions. This NADPH is to be important, and these to be special functions of functions that are associated with the RBC. So because if you see RBC where the major amount of NADPH is to be present, this is to be important for the maintenance of the RBC membrane. And second thing, the enough number of this reducing compound that present in RBC, so that can facilitate the binding of oxygen to the heme. And even that particular mechanism, anyway, I'm not going to discuss here, but you should be remember that these are the various kinds of the applications that are associated with the NADPH. Altogether, the significance which are under the pentoses and the NADPH. Clear? Then we can see the deficiencies. Is there any deficiency which is associated with this particular pathways? So definitely the deficiency, first deficiency, there are two deficiencies. One deficiency related with the glucose 6-phosphate dehydrogenase, we also call G6PD. So this G6PD deficiency, which is an inherited sex-linked trait. So you should be remember this is a sex-linked trait, and the deficiency will leads to the hemolytic anemia. So on hemolytic anemia mean there is a low amount of this hemoglobin that can present in this particular patients. And if you take certain drugs like anti-malarial drugs, antipyretic drugs, antibiotics will be involved for the patients, so that can the particular patients will be deficiency of this particular enzyme will leads to the hemolytic jaundice, right? Even not only that, even the flavonoids also be cause the hemolytic jaundice. This will call favism, right? These are the different kinds of the deficiency disorders that are associating with the glucose 6-phosphate. And have you ever think about this? Is there any advantage? So for suppose if you see this is an deficiency, definitely the deficiency mean always it should have a clinical symptoms, right? But does this deficiency will have any kind of advantages? So definitely you should be remember that not there are be few advantages which is related with these deficiencies, but here this is the appropriate time to discuss. The advantage for this deficiency is that this deficiency patients, they are resistant to malaria. So malaria, which is caused by the bacteria that is Plasmodium falciparum, this Plasmodium falciparum for its activity, it should depend on this pentose phosphate pathway, and it can try to reduce the glutathione reductase. So the glutathione reductase, because where exactly that can be function when more amount of NADPH present, but that NADPH can be utilized by this this malarial bacteria, so that less amount of glutathione peroxidase can be present, so that this particular bacteria is in this particular patient is not effectively grown, so there is no causing of malaria to that particular patients. The best example I have given the African people. The African people, they lack this glucose 6-phosphate dehydrogenase. Definitely they are resistant to malaria. So this is one kind of the adaptation to the climatic conditions. So in that particular nature, there will be a more amount of the mosquitoes, and that particular nature is predominantly involved with the more amount of mosquitoes. So this is one kind of the African adaptation, so that they cannot affected with this malarial bacteria. So definitely they are to be safe from the malaria. So this is to be one of the advantage related with this deficiency. And coming to the one more syndrome we call Vernick Karsakov syndrome. This particular syndrome is to be related with the trans ketolase activity. So if you see the trans ketolase activity, if you go back to the previous slide where I've shown the reactions where trans ketolase activity, which is important because for its activity it should require thiamine pyrophosphate. Clear? So it should require thiamine pyrophosphate. This thiamine pyrophosphate, because of this, there will be a few cases where exactly this TPP is not to associating with this trans ketolase. Because of this, the activity of the enzyme is to be reduced by tenfold. Clear? So this is related with the enzyme trans ketolase that is linked with the thiamine pyrophosphate. So this symptom includes mental disorders, loss of memory, and partial paralysis. Even the symptoms are manifested in alcoholics whose diets are vitamin deficients. The best example you can see here, the trans ketolase activity, which is measured in RBC, there that can be very low in the condition of pernicious anemia. So where exactly vitamin B12 will play the major role. It's clear? So these are the two different symptoms, disorders that are associated with the pentose phosphate pathway. It's clear, friends? Okay, then tomorrow we'll discuss about the next class.